Welcome to the Personalized Medicine Podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field. I am your host, Alexander Yahensky. Let's start. Welcome to episode 20 of the Personalized Medicine Podcast. I hope you are all safe and healthy in these challenging times. Wherever you are, remember to practice social distancing, wear a mask, and wash and disinfect your hands regularly. Sticking to those simple rules can help us overcome this pandemic. It's not an issue of one person or one country, it's the responsibility of all of us in the entire world to make this happen. Now, back to the podcast. I am very happy to present to you our new co-host, Aradhana Chopra. As you might know, Aradhana joined our team this summer and helped us behind the scenes to put the episodes together. Today is her debut on air. So give your heartiest welcome to Aradhana and join us for episode 20 of the podcast. It is a really good one. I hope you'll enjoy it.
Welcome to the episode 3 of season 2 of the Personalized Medicine Podcast. It is my pleasure to introduce our guest for today's episode, Dr. Cosimo Ducani, who is the CEO and founder of Moligo Technologies, a biotech company based out of Sweden that uses enzymatic methods to produce long and ultra-pure DNA strands at the industrial scale. Dr. Ducani completed his PhD in applied chemistry in 2011, where he received the prestigious Marie Curie Early Stage Research Fellowship that enabled him to work at University of Lecce, Italy, and University of Birmingham, UK. Following that, he moved to Stockholm and started his postdoc in Professor Björn Högberg's lab at Swedish Medical Nanoscience Center at Karolinska Institute to develop molecular tools, including the methods for enzymatic DNA synthesis. He developed the monoclonal stoichiometric MOSIC method for enzymatic DNA oligonucleotide production. In 2013, Dr. Ducani was awarded with a research grant from the Swedish Society for Medical Research and subsequently another grant from the Hagelin Foundation. Before starting Moligo Technologies, he received an exploratory pre-seed grant from Novo Nordisk Foundation, which is aimed to accelerate commercialization of research findings and development of novel technologies within the health sciences. Welcome to the podcast, Dr. Ducani.
Thank you. Thank you for having me.
I met you when I was doing an exchange at the Högberg lab during my PhD. As far as I remember, that time you were working on further improving the technique. Am I right?
Yes. At the time, I was working on optimizing the technology for enzymatic synthesis of DNA strands. As you know, most of the technologies to produce DNA, they are based on chemistry. In that case, I joined this project to develop a new method where you use instead bacteria and enzymes to produce these molecules.
Great. From there, you moved on to founding Moligo Technologies. And quite recently, the company has raised a seed investment to bring the enzymatic DNA technology to the market. Heartiest congratulations for that.
Thank you.
So, my first question, rather the first obvious question is, how did Moligo Technologies happen? Like, what motivated you to take the technique from the lab to the startup space?
Yeah, so basically at the time, a couple of years ago, I was still wondering whether I wanted to continue with academia, becoming a principal investigator, so a professor, or moving out of academia. I remember one day I was reading about a very interesting paper, which was published of Nature Biotechnologies, where they basically develop a technology based on enzymes to produce DNA oligonucleotides, and then they started the company out of this technology. It was quite different for what I was doing, but then I started digging a little bit more why they started a company based on enzymatic DNA synthesis, and whether there was a need of DNA made by enzymes. And then I realized that basically I had already a very strong, solid technology for producing DNA oligonucleotides, and there was a huge need of these molecules. But then I thought, okay, I really enjoy developing this technology. I wanted to, and also optimizing, and maybe it's time to actually bring in something to the people. And then I decided to start Moligo Technologies.
That's wonderful. That's really very nice. So the obvious, follow-up question would be, what does Moligo Technologies do?
Yeah. So basically, as I was mentioning before, usually DNA molecules, DNA strands are produced using chemistry. So this is basically but the molecules that are obtained by this approach, which is 60 years old, are very short, full of errors and low quantities. And well, this is a huge limitation with new technologies coming out, including gene editing, for example, diagnostics and DNA therapeutics. So what Moligo Technologies do is basically we develop a new technology which is able to produce ultrapure, so basically error-free DNA molecules, several thousand bases long and in industrial quantities. So, and we believe that with this technology, we can really help the new gene technologies, and we can bring them to clinical to industrial level.
So for our audience, you can find the link to Dr. Ducani's Nature paper on the Mosaic method and the company's webpage in our show notes. So, Cosimo, this technique is very unique and very interesting, because I can say that from the first-hand experience, because I also use this in my work. So, what are the areas where Moligo Technologies is targeting, like is focusing at the beginning?
So right now we are mostly focusing on gene editing, and because we believe that we want to produce donor templates for CRISPR-Cas or other gene editing technologies, because we know that there is a huge limitations in terms of the donor templates which are used for in these technologies. So usually the donor templates, either they are viral templates, which can be cytotoxic, and also they are full of errors. So right now we're focusing on this, but we want to expand in other areas like diagnostics, because usually we know, talking to our customers, our potential customers, that basically sometimes the impurities of the current DNA batches, it can be a problem during diagnostic processes. And then but the main goal, of course, because we are a large scale, we want to produce large scale production, is pharma company. So we want to produce also DNA therapeutics.
Oh, wonderful. So I think that brings me to my next question. How do you think Moligo Technologies can be helpful in the field of personalized medicine? And what could be its potential role in the nucleic acid-based vaccines? As we know that there are a lot of vaccine candidates these days which are coming up for corona and other viruses.
Yeah, that's a very good question. So when you think about personalized medicines, I mean, maybe you think about, of course, a pharma company who maybe have to develop a specific therapy for a single person, and that can be very, very expensive, of course, and that's quite difficult for companies. But we believe that thanks to our technology, we can bring many therapies, gene therapies and DNA therapeutics, to people by reducing the cost. So this is going to basically increase the chances of developing new personalized therapies for people. I think this is great. I mean, this is actually our main mission, to really bring these therapies to everyone, and with drastically reducing the cost. Because to me, there is not much sense to develop technologies in the end they cannot be used to people from people, which are not accessible. So that's why we were working in this direction. And to answer your question about the vaccines, definitely, I think Moligo Technologies has a huge potential in developing DNA vaccines. So, as you mentioned, there is this some of the vaccines which are coming out from, for example, Pfizer or Moderna, they are based on RNA. And RNA, it's a very unstable molecule, so it can be easily degraded. And of course, now you can see that these vaccines needs to be kept at -80 all the time. And this is a huge limitation if you want to bring this vaccine to poor countries as well. So definitely we want to also expand in this area by producing vaccines based on DNA, which is much more resistant, and it can be basically lyophilized and kept at room temperature without any problem.
We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, would like us to cover a specific topic, or recommend a guest, please write us an email to team@pmedcast.com. Or you can reach out to us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To download the show notes for this episode, visit our website, pmedcast.com. It's PMEDCAST.com. The show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. Make sure to check them out. And don't miss the next episode. Subscribe to our podcast on your favorite podcasting platform. Give us a rating and leave a comment. It will help us make this show better. And now, let's get back to the interview.
So my next question to you is, how is the startup space in Sweden like?
I would say great. I think Sweden is one of the best European countries in terms of startup space, and it's the Silicon Valley somehow of Europe, I would say. The very first advantage is something that here they call professor privilege, which means that as a scientist, whatever you develop, it belongs to you. So you have the right of the invention. So I think this is really promotes people to start companies. And then downstream, there is also a very good organization in terms of incubators. So, for example, Moligo Technologies is part of an amazing incubator at Karolinska Institutet Innovations. It's called Drive Incubator. There is a huge network of investors, but also foundations which do not support only basic science, but also startups. So I would say that is a very great environment an inspiring environment to start a company.
That's amazing. That sounds really inspiring. So, since you have seen both, best of both the worlds, and I would like to ask you that what are the similarities and differences of working in a startup and a lab? And if you have to pick a point from each that, you would like to take in or adapt in the other, what would that be?
Well, definitely, I would say that working for a startup probably is a little, of course, it's more product oriented. So we really work to solve short-term, or at least relatively short-term problems. While probably in a lab, most of the time you work on something which is interesting. It could be very useful, but you don't know when, if it will be useful in many years. So I would say this is the main difference, like while working in a lab and in a startup. Um, I don't know if there should be something actually, but probably from my experience, at least in my during my academic career, if I have to pick something from the startup to the lab, probably I would say that I would be a little bit more pragmatic without losing creativity, of course. I would probably cooperate a little bit more with with the teams I've been working on. Because of course in a startup this is crucial. I mean, you definitely are a team, and sometimes in academia, I mean, you feel that you have your own project, and you wanna learn, so you don't share as much as you could. But this is my personal experience, of course. Maybe this is not true for all the labs or all the people.
Okay, so now since you have, you have another feather in your cap, like previously you were Cosimo the scientist, now also you are Cosimo the CEO. So one piece of advice Cosimo the CEO will give to Cosimo the scientist and vice versa?
Hmm. Well, probably I would say that the CEO would tell the scientists that failure is part of the process, not to get too much frustrated about it, and not for too long, first of all. And well, maybe the scientist would say to the CEO to keep enjoying the science behind the business, because this is, I think, also the beauty of starting a company in life science.
That's really valuable piece of advice. That is really nice. So there are, I'm sure, a lot of our listeners are budding or aspiring entrepreneurs from different spheres of their career, like from PhDs to postdocs. Any tips for such aspiring entrepreneurs you would like to give who want to venture into the life science startup space?
Well, definitely. I think if you are a PhD, if you are a postdoc, and you have a great idea, you think that you really believe that you have something that you can bring into people, and you can help some of the areas you're working in, you should definitely try out. I mean, I could see that some people maybe are afraid of instability, because I mean, you are a startup, and the first thing they tell you when you start a company is the rate of startups which fails. But we shouldn't be afraid of just starting something from our ideas, because in the end, I mean, we would regret more of not starting rather than failing. So I would definitely say that try to talk to people. Try to, if you have a technology, sometimes if you don't talk to people around you, maybe you don't know that your technology can be useful. I can tell you that some maybe analysts of some investors in the past, they ask me who cares about ultra pure long DNA. And then if you really talk to customers or you talk to people, you see that maybe there is a huge need. So really be brave and try to talk to people. And then if you want to start a journey, maybe find also the right team, because in my case, the team is really supportive, and we enjoy together basically working and bringing this product to the market.
Wonderful. So, like running your own company, and so are there times where you miss working on the bench, or do you still get time to work on the bench?
Uh, Recently, I didn't work much on the bench, unfortunately, but yes, I miss a little bit. Of course, we are still a small team, and so I mean, we need to somehow also help each other in the different tasks. Sometimes I try to help in this, and to do some science as well. And the thing that probably I miss from science, so you know very well that most of the experiments, I mean, do not work usually. So, and you will live for the small moments when something works, and I miss this eureka moment, let's say, in science. So that's maybe the only thing I really miss, I would say. But I have to say that I have talented people in my team, scientists, that they are very willing to share these moments with me. So I cannot complain about it.
That's great. You have a very nice and supportive team. I think one but last question is, where would you like to see Moligo Technologies in the next 10 years?
Well, definitely, I think Moligo Technologies, in my opinion, it really can change life science in total. So I would say that I would like to see Moligo Technologies in many fields, from agriculture to therapies to bioelectronics and diagnostics. So I really believe that DNA, it's a very programmable, easy molecules, if we can say that, but with a huge potential in so many fields. And the fact that we can also make these molecules, besides ultrapure and long, we can also attach some functionality, so we can even tune the properties of these molecules. Then the only limit is imagination, I would say.
Great, absolutely. So, Cosimo, it was a really nice talking to you today about how you started Moligo and what are the future plans of Moligo. And I had a really wonderful time. But before we let you go, could you tell us that how can our audience reach you online?
Well, if you go on our website, www.moligotechnologies.com, then you can find my email. So, or on LinkedIn, you can find me. You can reach me out, definitely.
Great. I'm sure there'll be a lot of people, a lot of aspiring entrepreneurs who would like to ask you a lot of questions, and they will definitely reach out to you. It was again a pleasure talking to you today, Cosimo, and thank you so much for taking out the time to talk to us.
Thank you. Thank you. It was my pleasure. Thank you very much.
Thank you so much for being with us today on the Personalized Medicine podcast. If you like this show and know someone who would enjoy it too, please share this podcast with them. And don't miss the next episode yourself. Subscribe to the Personalized Medicine podcast on your favorite podcasting app. You can find us on Apple Podcasts, Spotify, Pocket Casts, Stitcher, iHeartRadio, and many, many more. Please rate us there and leave a comment. That helps us to grow and deliver the best experience to you. To access the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. And engage with us on social media, where we regularly share the news and exciting content on personalized medicine. You can find us on Twitter, LinkedIn, and Facebook just by typing in Personalized Medicine Podcast, or use our handle, pmedcast. And if you have any feedback or would like to suggest a guest for the show, write us an email to team@pmedcast.com. Have a great day, and until next time.